Ladies and gentlemen, good afternoon and good day. Welcome to Sydney. Delighted it's finally turned on some weather that can be appreciated, and more to what is hopefully normally Sydney weather at this time of the year. We're delighted that you could all attend. For those who don't know me, it is my honour to be the President of Motor Neurone Disease Australia, and my name is Ralph Warren. This session has been repeated earlier in previous Alliance meetings, and it's been proven to be a wonderful success, both in an exchange of ideas and views and knowledge from our experts that we assemble and from your personal experiences that you generate and you forward to us by way of questions. Professor Dominic Rowe, who will be chairing this afternoon's session, will introduce each individual speaker more fully. But I'd like to thank all of the speakers that are going to be with us this afternoon. They are experts in their field. They come with great knowledge, and we personally thank them for their time they've spent in coming here and sharing their knowledge with us. When I'm on the thank yous, could I also indicate and thank the Commonwealth Government of Australia for the contribution they have made to assisting people living with motor neurone disease and their carers in attending here today and also in attending online. This is a new innovation that we have had this year, and we hope and we earnestly believe that it will be of great benefit to those who can't afford or cannot afford the time or capacity to attend here in person, and we welcome them also. When you are standing or delivering a question, could you please make note and take account of, we have a number of questions to move through this afternoon after the speakers have given their short deliveries. The more questions we get, the more knowledge that can be imparted to you. Please keep your questions succinct and to the point, and then that will assist all people to get as much knowledge as we can, both online and in person this afternoon. I'm advised that Anna Rogers, a filmmaker from Independent Pictures, is filming this session this afternoon. The filming will be largely as a result of where the camera is positioned from the rear. If you have any difficulties with being inadvertently or advertently involved in the filming, please let us know, and we'll do what we can to attend to any problems you might have. So, thank you for your attendance. It promises to be a most fruitful and enlightening afternoon, and I call on Dominic Rowe, who is chairing this afternoon's session, to introduce the speakers and commence. Thank you, Dominic.
Thanks, Ralph. Good afternoon. We have a fair bit to get through this afternoon. I'd like to actually run with each of the speakers talking for 20 minutes, and then we'll do our questions all together. So even if you have a burning question, we'll try and do that all together in the question session. It gives me great pleasure to introduce these four speakers who've come from Ireland, England and North America. To introduce first, I'd like to ask Kevin Talbot to come and talk about what do we understand about motor neurone disease compared to 5 to 10 years ago. Kevin is a professor of motor neuron biology at Oxford. He runs the Oxford Motor Neurone Disease Care and Research Centre, and he's going to enlighten us this afternoon. Thanks, Kevin.
Thanks very much, Dominic. It's a tremendous pleasure to be here in Sydney and to come and speak to you today. And I think my job is to try and set the scene. Some of the things I'll mention in passing will be covered in much more detail by subsequent speakers. I think it's probably reasonable just to make sure that we all understand exactly what we're dealing with motor neuron disease. it's a disease of a very complicated system. Here you can see that if we want to make a voluntary movement, some cells at the top of our brain receive a lot of complicated inputs from elsewhere in the brain and descend as a large bundle of fibers through the brain and into the spinal cord. And that's what we call the upper motor neuron pathway. And it's highly complicated. It's evolved very rapidly in recent evolution. Humans have a rather special kind of system for voluntary movement. And then down in the spinal cord, that very spidery cell that you can see there is the lower motor neuron. And that, again, it has many, many connections with the local cells and other cells. It's a very complex piece of machinery. And the green and red picture there is the neuromuscular junction, actually, of a mouse from our laboratory. And it shows that each of these motor neurons in the spinal cord has a very long process that goes out into the limb. It's incredibly long. It's as if I was standing here and the end of my cell were 30 kilometers away. So how that cell functions in the first place is still something of a mystery. But what we do know is that it goes wrong. And it goes wrong in motor neuron disease or amyotrophic lateral sclerosis in a rather dramatic way. And the picture at the bottom is cells from the spinal cord of a patient who died of motor neuron disease. And what you can see there is that there is protein that is misfolded and aggregated, and that's stained brown. There are currently almost 5,000 people in the UK living with motor neuron disease, more than 20,000 in the US, and 1,500 in Australia. And each person in the population probably has a risk over their whole lifetime of about 1 in 500 of getting motor neuron disease. So it is not as uncommon as sometimes people think. The key thing that we do know about motor neuron disease, and which there's been some very good work in the last 5 or 10 years, is the epidemiology of aging. This is a condition which gets commoner as you get older, and that's a key fact. If we want to understand why motor neuron disease occurs at all, we have to understand what happens to the aging brain. It's also a very complicated condition clinically. Once you start seeing lots and lots of patients with motor neuron disease, you begin to realize not every patient is the same, and every person is on their sort of individual journey with this very difficult condition. And this is some work done in Italy looking at a population study to ask the question, can you actually find different patterns of disease, and what does that actually mean? And it's quite clear that in clinic it helps us very much to advise people about the kind of journey that they are taking with this disease compared to other patients. That's very, very important when often the source of information that you get is rather generalized and not very individual. Another level of complexity comes when you start looking at the brain and spinal cord after people have died. And these are 4 pictures all showing motor neurons, but they look quite different. So people who carry a mutation in SOD1 have quite a different appearance to people who have more classical ALS, if you like, which stains with a protein called TDP43. There's also a protein called FUS, which occurs again in a distinct group of patients with MND. And I think Bob Brown is going to tell you more about this in detail, in particular about a most recent exciting discovery of a new genetic form of motor neuron disease. But my point here is that this is not one disease pathologically. It's not one disease clinically. And if we've learned anything over the last decade, it's that this level of complexity has to be understood if we really want to try and find some proper therapies. So if you take motor neuron disease, you can say the same thing about it as you can say for Parkinson's disease and Alzheimer's disease, which are the other common forms of neurodegeneration. The problem we have in trying to really conquer these diseases therapeutically is when patients arrive in the clinic, they've already had the disease for a considerable period of time. There's diagnostic delay, and that's something we really have to overcome. There's a high level of complexity, as I've explained to you, and actually, although we're beginning to make very significant advances, we still have a very incomplete understanding of the basic biology. And importantly, we have really a lack of disease markers. You have motor neuron disease because an expert in neurology tells you you do, and then we only really have rather crude measures about how to actually see how people are progressing, which makes therapeutic trials very difficult. Another problem is really understanding when the disease begins. This graph here is simply an idea. Is this in the blue line a disease where over many, many years motor neurons degenerate very gradually and you reach a threshold and then you develop weakness when you get down to a certain level? Or is it a condition where the motor system is completely normal for most of your life and then really in a very short space of time things start to unravel? We don't really know which of those 2 models is correct, but what we do know is that the clinical phase is preceded by a period when people are susceptible. They have some sort of susceptibility profile which primes their nervous system. Then a preclinical phase, we have no idea how long that is, where things are starting to become stressed, things are starting not to work, but the patient doesn't notice that. And then they start developing weakness. Now, at the moment, the purpose of this slide really is to try and explain that the clinical trials that we're performing at the moment are all being done in this clinical phase, obviously, where patients have established disease, and often, on average, they've had the disease for at least a year before they come to our clinics. And this raises major problems because we'd really like to roll that back so that we can start working in a phase of disease where people have the earliest possible signs of disease. So we desperately need to be able to do that, and that's very challenging. So there's a gap between the number of drugs that have been tried in motor neuron disease, and that's just a very incomplete list, and the number that have worked. So we have a very large amount of advance, some of which you'll hear about in subsequent talks, in basic science, and this is genuinely exciting. But why have we not managed yet to turn that into treatment? That's a question that most people living with the disease today really want to know the answer to. So how do we get over that translational gap? As I've mentioned, we have to understand the basic process. Who gets motor neuron disease? When does it start? And why does it start at that moment in time? If you've been essentially healthy for a long period of time, why is it that the motor system starts dysfunctioning? How do the cells die? What are the mechanisms that we can target with drugs? Secondly, we need some way of monitoring the disease. We need what are called biomarkers. We need a way of taking small groups of patients, rapidly screening them so that we can see whether drugs are having a biological effect, and then taking those drugs into larger clinical trials. At the present time, we need 500 patients in each arm of a clinical trial, maybe 800 to 1,000 patients to demonstrate whether a drug works or not. That's extremely expensive, it's time-consuming, and it's very, very difficult. We need a way of improving that. We also need to really target the earliest possible changes. And we need better models in the laboratory to do that. And I think you're going to hear about some very exciting models to do with human stem cells in one of the subsequent talks. But actually, there is still a place for using mice and other forms of tissue and model in the laboratory. So what progress have we made in those areas over the last 10 years? Now, this is a complicated slide, and I don't wish you to understand every element of it, but what it's trying to say is that we have grossly underestimated the genetic contribution to motor neuron disease. Maybe some of us have, some of us haven't, but 10 years ago, it was clear there was at least one gene which, if it was mutated, led to motor neuron disease. There were people who had familial disease. And now, as Bob Brown will tell you in one of the subsequent talks, we know a lot more about the genetics of motor neuron disease. But what we also know is that there is a significant genetic contribution to what's called sporadic disease, so people who don't have a family history and who don't necessarily have a significant chance of passing that disease on, but the way that they are made up genetically is one of the most important reasons why they are susceptible. And we now have the technology to understand that because of very high throughput genetic approaches. So you can actually take somebody's DNA and you can screen each individual gene and have a look and see what the variation is. Actually, what you get back is a vast amount of data and a lot of complexity. But if you can use that with some very sophisticated analysis of the way that people actually appear in your clinic, the graph I showed you right at the beginning, you can start to unravel why some people behave in one way, some people behave another way. And so those significant and strong genetic effects, I think we're very close to really analyzing those in great detail. Genome-wide association studies use another kind of technology to look at the variation across many, many genes which have common variants. So they exist out there in the population, but they're slightly commoner in people with motor neuron disease. Why is that? How does that make you susceptible to the disease? Combining those two things together over the next 5 to 10 years is absolutely going to give us some information we didn't previously have. So, as I mentioned, the SOD1 gene has been known about for almost 20 years, and now we have probably 75% of the genetic explanation of familial disease. And that's giving us a way into pathways that are disturbed in the disease and that can be genuine targets for therapy. However, we've had that model of motor neuron disease based upon SOD for a long time now, and we haven't managed so far. What we've got is a lot of information about this model, and it's given us lots of clues about the pathways that are affected. So there is no shortage of information. We know that the cells in your nervous system, the motor neurons, rely on the contact with other cells called glia, which nourish them and support them. We know that these cells connect with muscle, and we know that these tissues are probably very important because this is a system disease. It's not just one cell that's degenerating. We know that inflammation might be important, that mitochondria, which are little batteries inside your cells which provide energy, that they're very vulnerable in people with motor neuron disease. We know that various types of cellular stress associated with aging are relevant. So we have a huge amount of information. And the question is why that has not been translated into drugs. And it's probably because we're still uncertain what is the cause, what is the effect. So which of these things that we've been studying is a consequence of a disease and which actually are involved in its causation. So it's back to what I said earlier about understanding the very early pathways. We need to know who is vulnerable out there. How can we identify those people early in life? How can we study them? How can we try and treat the disease before it starts, really? So I've mentioned that aging is very important, and I've mentioned that genetic variation is very important. Those two things are absolutely clear, and there's no doubt about them. What about the environment? It makes common sense. Every patient that comes to see me asks the question, what is it about their occupation or what they've eaten, where they've come from, where they live? Are these factors relevant in their disease? And it's a very reasonable question. Actually, we have very little solid information that the environment makes any difference at all. If it does, it's probably very complex, and it's probably very different in different people. That's just a graph of smoking, which has been studied over many years, and there's conflicting evidence, but it seems that maybe smoking gives you a very, very slightly increased risk, but it's extremely small. What about fitness? You may have heard that there seemed to be an excess of people who have a sporting background. That's a picture of Lou Gehrig, the American baseball player. And it's very clear to those who sit in clinic and see patients with MND that they're not necessarily the same as the rest of the population. Frequently what you hear is, I've always been so healthy, I've never had any other illnesses. So we've tried to study this objectively, if you like, by using the NHS system in the UK has a record linkage system. So you can ask the question, if you take people who've had heart attacks, are they more or less likely to have motor neuron disease? And in fact, they seem to be people who have motor neuron disease seem to have a lower risk of other diseases. They seem, if you like, to be fitter. Now, some people have interpreted that as meaning that doing the exercise gives you motor neuron disease. And while we don't know the answer, I think that's extremely unlikely, personally. I think that it's probably the fact that you're fitter means you have some genetic susceptibility, something that is very valuable to us as a human species early in life, but as you get older, carries a sort of penalty. But this is something we have to study in more detail. So what I've been telling you then is that motor neuron disease is not a simple condition with a single cause and almost certainly will not have a single treatment. It's a complex endpoint of many different processes, some of which are strong genetic effects, some of which are complex genetic effects, the aging process and possibly the environment. And they all come together to cause this cellular change. And we need to understand this extremely complex puzzle. So the mouse problem is, that we are different from mice. And so people have tended to, because mice live for 2 years, create mice that get the disease in a very short space of time. And that approach, I think, has its limitations, because what you tend to do is to create some false leads, if you like. So what we're trying to do is we've created a mouse where the human gene, with all of its regulatory sequences, is put into a mouse and is expressing it at very low levels. And we hope that that sort of approach will allow us to actually create a mouse where we can look at those very, very early pathways that activate disease. The biomarkers I mentioned to you, so this is just examples from my own work, but other groups are doing the same sort of thing. We're studying patients longitudinally, so we're taking people every 6 months who are very kindly offering to have a lumbar puncture and an MRI scan. And we now have, halfway through this study, we have a lot of data on what's happening to people as the disease develops. And I think this kind of study is going to be critical for really understanding what's actually going on inside people's nervous system. So what I believe very firmly is that good care and good research go together. And I don't think that I could do the sort of research I do unless I also saw patients in clinic, because that helps me understand the disease, but it also allows me to enter into a partnership with my patients who very much are a key partner in the research. So we see about 150 new patients a year, and we offer them regular monitored care with planning and intervention in nutrition and breathing at an early stage. And there we hope to avoid crisis management. And patients are very keen to become involved in our research. And actually, the research into the care side of things, I think, has been a major success story in the last 10 years. And here is a study from Newcastle in the UK showing that non-invasive ventilation, which intuitively seems like a reasonable thing to do, but there's clear evidence that it works. It can give people good symptom control. It can also increase life expectancy. Now, this is the sort of study that some people might have thought, we don't need to do this because it's so obvious. But actually, this paper, certainly in the UK, which is a very heavily regulated healthcare system, this paper allowed us to go to the government and say, this is something we have to do. And so now people in the UK, compared to 10 years ago when I first started my clinic, where it was a real struggle to get access to non-invasive ventilation, now people have clear and free access to that. It's accepted as a standard treatment. So this kind of care-based research is absolutely critical. We need much more of it. So in summary, then, I think that there's a huge amount of advance in basic science. It's the kind of thing you always hear people saying, but I really do believe this, that I think we're in a golden era of understanding the nervous system and understanding neurodegeneration, largely because we have the tools to do it now. And this is going to lead to a lot of information. The question is how to translate that information into new treatments for patients. And we need new models. We need better ways of studying patients. The patients are the best model of the disease. And in the meantime, we need to continue to try and develop the best possible care, to disseminate the idea that multidisciplinary care is the right thing to do. We need to provide an evidence base for that. And there are increasing numbers of drug trials, and we need to do those in a more sophisticated and efficient way. So I think we will get there. This is a technical problem. It's one that's going to be solved, and we'll only get there in partnership with our patients. Thank you.
Thanks, Kevin. That was fantastic. I'd like to call